Thank you for the introduction. Good morning, ladies and gentlemen. My name is Tetsuichi Saito from the University of Pittsburgh. It is a great honor for us to have the opportunity to present our work here, and I would like to thank the ICS, my mentor, Professor Yoshimura, and all my colleagues. So as an introduction, detrusor sphincter dyssynergia, DSD, induced by spinal cord injury, rostral to the lumbosacral level, leads to inefficient voiding with high post-void residual volume. Previous studies show that the C-fiber targeting treatments, such as anti-NGF or capsaicin pretreatment, reduce detrusor overactivity. However, these treatments did not show any improvement of inefficient voiding or DSD. From these results, we hypothesized that the A-delta fiber bladder afferent pathways are involved in lower urinary tract dysfunction after spinal cord injury to induce inefficient voiding and DSD. So, the aim of this study was to develop a new therapeutic modality targeting A-delta fiber bladder afferents and to confirm that the A-delta fiber targeting treatment can improve DSD. To treat A-delta fibers, we focused on a drug called Ivermectin. This drug has been approved by the FDA for parasitic infections. It also activates a chloride flux in neurons expressing exogenous glutamate-gated chloride channel receptors, thereby shunting excitatory impulses causing hyperpolarization. However, the problem is that these receptors are not present in humans or rodents. To use ivermectin, there's a glycine receptor with a double mutation, which is derived from a human receptor, and it has a larger single channel conductance, and the sensitivity to ivermectin is almost 100 times higher than wild-type glycine receptors. By using this glycine receptor, we only have to use 1% of ivermectin of the normal dose. So this G2M receptor genes were inserted into replication-deficient herpes simplex virus vectors. This is an explanation of how we target the A-delta fiber. By selecting a HSB vector driven by a subpopulation-specific promoter that can only duplicate in cells that expresses neurofilaments, the G2M can be delivered specifically to A-delta fiber bladder afferent neurons. In this study, neurofilament 200 promoter-driven HSB vectors were chosen to target neurofilament-expressing A-fiber neurons in bladder afferent pathways. This is the study design for morphological analysis. The spinal cord was transected on the level TH89. 2 weeks after spinal cord injury, M-cherry, which is a fluorescence marker delivering vector injection, was done inside the bladder wall. 2 weeks after injection, the DRG of the LG-S1 was harvested and histology was performed. Another set of an experiment was designed. The spinal cord injury was done as I previously explained. 2 weeks after spinal cord injury, mutant glycine receptor delivering vector injection was done in the bladder wall. 1 week after bladder injection, 1 micromolar of ivermectin, IP, once a day was done for 7 continuous days. After 7 days, CMG, EMG was performed, and the DRG was harvested for PCR. This is the results of the histology of the DRG sections. As you can see, positive M-cherry staining in neurofilament expressing bladder afferent neurons can be confirmed in the red high spots. And the area size of the M-cherry positive DRG neurons significantly increased in spinal cord injury compared to spinal intact mice. This is the result of a single filling cystometry under awake condition. In spinal cord injury mice with G2M, ivermectin treatment did not improve detrusor overactivity shown by non-voiding contractions during the storage phase. This is the results of the EMG after awake condition. So the chart on the left upper side is a typical EMG-CMG chart we see in the spinal cord injury. During the voiding contraction, you do see a lot of EMG activity and a little bit of EMG reduction. During these reduction times, the mice void. However, as you can see in the green arrow sections, you don't see any bladder pressure decrease. And at these points, they do not urinate. They do not have any urination. However, as you can see on these red arrow spots, you do see a big pressure drop, which causes urination events. So what we did is we calculated the EMG reduction, which causes these big notches in this voiding contraction. And when you compare spinal cord injury and ivermectin treatment groups, as you can see in spinal cord injury, you do see some notches. But with treatment of ivermectin, the number and the area of the notches increase significantly, which leads to percent ratio of the EMG activity reduction time increase and leading to reduction of the voiding efficiency. These are the results of the DRG, PCR of the DRG. In spinal cord injury, G2M, spinal cord injury mice with G2M, ivermectin treatment reduced the messenger RNA expression of ASIC1-3 and piezo2, which are known to act as mechanosensitive channels in the DRG, but not TRPV1 that is predominantly expressed in the C fiber afferents. So as summary of the results, histological studies show that cell area size of DRG neurons was increased in spinal cord injury mice, suggesting that the plasticity of the A-delta fiber bladder afferent neurons could contribute to the enhancement of bladder to EUS reflex activity, resulting in DSD after spinal cord injury. Ivermectin-induced activation of the double mutant glycine receptors expressed in A-delta fiber bladder afferent neurons improved DSD, evident as increased voiding efficiency and EMG activity reduction time during voiding bladder contraction. Our PCR of the DRG shows significant reductions of the A-delta fiber-related mechanosensitive channels with G2M and ivermectin treatment, whereas C fiber-related channel expressions was not altered. As conclusion, gene delivery of G2M designer receptors to A-delta fiber bladder afferent neurons using HSV vectors with subpopulation-specific promoters such as neurofilament 200 show that A-delta fiber bladder afferents contributed to the EUS hyperactivity in DSD after spinal cord injury. G2M receptor delivery enables to develop a druggable approach using an exogenously applied synthetic ligand, ivermectin, to suppress A-delta fiber bladder afferent activity to improve DSD and inefficient voiding in spinal cord injury mice. Thank you for your attention, and I'm open for questions. Thank you.
Did you hear anything of what I said? Well, it wasn't that important anyway. I start discussion. Chris Fry.
Very nice talk, thank you. Can you be sure that it's the channel is only at the on A-delta fibers? Could it be in any other tissues in the bladder wall? Have you checked in other tissues in the bladder wall?
Just to confirm, are you asking whether they are in the bladder wall?
Or in the ganglion itself? Do you know where, if it's exclusively where you suggest it is?
Actually, this virus can only suppress in neurofilament fibers, which, well, from the bladder, there are A-delta fiber and unmyelinated C fibers, which we suppose that is expressed only in the A-delta fiber.
Okay. So you, I mean, I don't want just to press. I mean, have you looked at the nerve fibers in the bladder wall to see if it's expressed there or not?
In different experience we have, but not in this experiment.
Not, okay, all right, thank you.
More questions? Well, I have a small question. Okay.
Yeah, I would like to know, did you weigh the bladders?
The bladder weight?
Yeah.
Yes, we did. In spinal cord injury, they were significantly heavier than spinal injury.
Okay. And the increased size of the dorsal root ganglion cells, could that be due to the increased size of the bladder rather than of the lesion of the spinal cord?
So, my personal thoughts. Well, in former studies, like in the past, there are studies talking about brew models increasing NGF, which causes brew models, which causes bladder hypertrophy, which leads to increased NGF and increases the DRG cell sizes. So I suppose that everything might start from the bladder hypertrophy, maybe leading to increased BDNF, which leads us to the DRG cells increase. So I do not know which comes first, the hypertrophy or the increase of BDNF, NGF, but I suppose that there is a relation between the bladder size increase.
Okay, I have to put it on. You write that you treat these animals with compression of the abdomen to empty the bladder once a day. Once a day, well, the normal mouse voids at least 20 times a day. Could this influence a little your morphological findings and bladder hypertrophy and so on? And how could that influence what you present as your results?
Yes, our group has previously studied the difference between like 1 time a day, 2 squeezes a day, 3 squeezes, 4 threes, and it has been published in the neurodynamic and neurology in the past. And we do see a lot of morphological changes, a lot of changes in the trip channels. And I suppose that 1 time, and from the results, 1 time a day is more sufficient for like a more overactive bladder model. But for a DSD model, we have not confirmed that yet. But yes, there is a morphological difference between 1 time, how many times we squeeze a day.
Okay. So it doesn't actually do very much then because you have controls for it. What do you say, Naoki, about this? Because I think this is highly unphysiological if you want to state
so
that.
that's the way I try to maximize that the bladder dysfunction in this model. So we, as he said, compared with that multiple squeezing to reduce the. So this type of overactivity, or you said, it's actually related to fast over distension of a bladder due to that DSD or this type of that absolute obstruction. So then this DSD over distension cause a hypertrophy, overproduction of NGF or other growth factor like a BDNF, which affects that afferent pathway to induce hyperexcitability, to induce due to the overactivity, maybe exaggerate this DSD condition. So we use a kind of extreme condition to induce that maximal bladder dysfunction in this model. But as you said, so if you control the overdistension a little bit less, so this type of that overactivity or bladder dysfunction could be less to be developed. Even this extreme hyperactivity or DSD condition, this treatment targeting A fibers to activation of these mutant receptors improve this condition. So this is a kind of proof of concept. This type of study is working in this situation, even it's a very extreme lower unit tract dysfunction in this model by using that just 1 time day squeezing of the bladder.
Thank you very much.